So hopefully all of you are getting my voice.
Yes, sir.
Thank you so much. So we are starting the workshop, and so before start, I want to mention some things which are I want to tell you before start of this workshop. So generally, this workshop is carried out under the banner of BS Council Education and Research, which is founded by the Dr. B.S. Kuchekar. And I just welcome all the dignitaries, then esteemed colleagues and distinguished guests for this workshop. And welcome to our molecular docking workshop. It's immense pleasure to host this event under the esteemed banner of BS Council of Education and Research, uh, which is a foundation dedicated to empowering education, elevating academically gifted yet financially constrained students. Before we dive into the intricacies of this molecular docking workshop, let me allow a moment to share the something that is close to our hearts. The entirely of these proceedings means whatever the amount I get from this workshop, that will be generously donated to the BS Council. And this gesture is a testament of our commitment to not only foster scientific inquiry, but also to support the noble cause of education and research. At the helm of this BS Council vision uh, is Dr. B.S. Kuchekar, a luminary in the field of pharmacy, uh, whose career span over 4 decades. Dr. B.S. Kuchekar is not only a principal professor of pharmaceutical chemistry at the Maharashtra Institute of Pharmacy, Pune, but also he is a beacon of a hope of all of us many. His academic journey is adorned with an accolade, including Bachelor of Pharmacy, a Master of Pharmacy with a gold medal, a doctorate, Bachelor of Laws, and postgraduate diploma in patent law. His teaching stage began at Pune College of Pharmacy and has since impacted countless lives throughout his role as various institutions and now as a senior professor at the MIT Pune. So Dr. Kuchekar dedication to education and research in the pharmaceutical sciences is unparalleled. His journey from the student facing numerous challenges to leader in the education is a source of our inspiration to all of us. And without further ado, I would like to invite Dr. Kuchekar to share few words that illuminates the path of knowledge and dedication that leads us forward. Sir, please.
Thank you. Thank you, Dr. Somadatta. Dr. Somadatta Chaudhary, all the participants to this esteemed workshop. Friends, Dr. Somadatta Chaudhary has conducted this workshop. This is the 20th batch of this. About more than 1,000 candidates have trained through this workshop successfully. When I met Dr. Chaudhary, I narrated about the establishment of BS Council of Education and Research, which is dedicated to help the poor students and academically strong students, meaning talent and poor students. My friends, immediately he told me, sir, I am conducting such type of workshop. Whatever may be the amount generated through these workshops will be given to BS Council of Education and Research. That is nothing but towards the poor and talent students. Thank you, Dr. Somadatta Chaudhary. And I would like to tell you within a 3, 4, 5 minutes about this BS Council of Education and Research. This is actually a non-profit organization and established in August, but actually operation started in December. Within 3 months of span, sir, we have assisted 9 beneficiaries.
Wow.
The amount
Great,
may
sir.
be from 15, 21,000, 25,000 like this. But in near future, we are going to help. We are going to pay the full fee of the needy students, sir. Now, it is 3 months age of this BS Council of Education and Research.
Very nice, sir.
See, Dr. Somadatta Chaudhary has immediately told me, sir, I will definitely help to for this. My friends, your participation, your contribution in this particular workshop will be used to help the poor and needy academically strong students. See, this base council of education and research is established, and also it has a legal existence. Legally, it is incorporated. We have the approval of Niti Aayog also. We have ATZ tax certificate also, and in that case, we have not even advertised in social media also, but for this conference, sir, I got so many calls, but I told those principals and the lecturers that Dr. Somadatta Chaudhary is insisted I have to train the students one to one. I don't want to increase the number. Next thing which I would like to tell about Base Council of Education and Research is even you participants can become the part of Base Council of Education and Research. BS Council of Education and Research, that is BSCR. This is not the property of BS Kuchekar. All of you are involved in it. Even if you want to help as a volunteer, or if you want to conduct some activities so as to raise the funds to help the needy students, you can always welcome. And for detail about Base Council of Education and Research, you can go through the website bscer.org. You will get every detail of this my council, BS Council of Education and Research. Thank you so much, and I wish you all the best for these participants. I think you will take many more knowledge about this molecular docking, and in future you will also train such type of participants in future. Thank you, Somadatta Chaudhary, for conducting this particular workshop for the benefit of BSCR. That is BS, not BSCR, but for the benefit of the needy students, poor students, as well as academically strong students. Thank you so much.
Thank you so much, sir, for these enlightening words and for setting the stage for today's workshop with such a grace and wisdom. I extend my heartfelt gratitude to all of you for participating in this workshop. Your presence and contribution are what make this event not just gathering of minds, but a collective effort towards making a significant difference in the lives of many deserving students. So let us now proceed with our workshop, keeping in mind that dual purpose of our gathering today. So to advance our scientific understanding and to contribute to a cause that support education and research for those in need. Thank you once again for being a part of this workshop, and let us make this workshop memorable and impactful event. Thank you so much, sir. So due
Thank you.
respect with the sir, I'm going to start this workshop. So generally on this slide, see, before going to molecular docking, I want to tell you some what are the importance and whatever the applications of a computer-aided drug design for the design of a new molecular entity. So in generally the old approach, the which is a trial and error method, in which we are going to identify the novel compound on the basis of a random screening, trial and error method, then ethnopharmacological approach, then serendipity method, then classical pharmacology. So these are the some basic research by which there may be a chances of pitfalls during the process of a drug design. But if you go for actual if you go for application of this computer aided drug design approach, which is the modern drug discovery, so it is like a modern technique which we are using to find a way for the destination, like that of a GPS. So it is a metaphor of a modern tools aiding in a precise measurement and prediction in the drug design, that akins to digital meter giving accurate reading, representing an accuracy and precision of a computer-aided drug design. So if we apply these computer-aided drug design approaches to the older approach, then definitely we can focus on screening as well as a rational design, as well as if it's we can get a help of its efficiency as well as the precision in designing the new novel compounds. So drug design is a conventional that slide which is related to the conventional drug design approach, in which the basic research generally it takes over 3 to 6 years, a wide net in cost of cast with the 5,000 to 10,000 molecules which we are going to assess. And then after that the drug discovery research, which is the phase in which we can narrow down these molecules into 250 promising candidates. Then developmental research, which is in which again we narrow down these 250 molecules to the 5 compounds only, which with extensive research which is conducted over near about 6 to 7 years. Then the clinical research, which is in which we are going to do the human trials commences. Then we are scaling this from 20 to 100 volunteer in the phase 1 to over 500 in the phase 3. So after that there is a market survey, market authorization and post-marketing research it is there. So after successful trial, the drug is approved and monitored in the market. The entire journey of this drug design older approach is approximately of about 13 years long and can cost around $2 billion. So emphasizing the resource and intensive nature of this to get a novel compound to bring to the market from the research. So where we can apply the computer-aided drug design approach in the drug discovery phases. So this the graphics, which is generally related to the interplay between the various phases of a drug discovery and computer-aided drug design strategies. So starting with the disease-related genomics, it progresses through the target identification and target validation utilizing the bioinformatics and computer-aided drug design. Then lead discovery that leverages techniques like library design and pharmacophore modeling, while lead optimization uses the QSAR and structure-based optimization. Before entering into the clinical trial, preclinical tests are done that incorporates ADMET prediction and pharmacokinetic simulations, which all are underpinned by the computer-aided drug design strategies to streamline and enhance the drug development process. So the image, the graphics that you are seeing on screen, that image that conveys the concept of a drug discovery using the metaphor. A database of a keys, generally it is a there may be a potential drug in the search to find the right key, that is nothing but a drug molecule that fits a specific lock, which is nothing but our receptor to treat the disease. So the right key represent the successful drug candidate that will unlock the therapeutic potential by interacting effectively with the biological target. So before going to this computer-aided, any type of a computer-aided drug design process that is totally related to the protein first. if you are aware about the protein structure, then and definitely you will get to the know the whatever the effect of that particular drug onto the protein structure. So if you are aware about the structure of protein, you are at 50 per, you can start your research, and you get a knowledge of about 50% of your research study. So what is a protein? As we are studied in a B pharmacy, there is a one subject that is a biochemistry. So that is related to the structure of a protein. So it contains the protein structure contains the four parts. Generally the first one, it contains a different levels like a primary, secondary, tertiary, and quaternary, which depict the primary. So for example, primary structure, it is generally a linear sequence of amino acid, and the secondary structure, it is nothing but it is a localized arrangement of alpha helix and beta sheets. So alpha helix and beta sheet, they are nothing but a form due to the interaction, intramolecular and intermolecular interaction between the amino acid which are present in the linear chain of a primary structure. So these secondary structures are the backbone of a protein. So then again, those are the remaining part of this from this primary structure, that is called as a loop region. So alpha helix, beta sheet, if they are generally joined together with the help of, see, this is alpha helix and this is a beta sheet, and this is a this is the primary structure. So when there is a interaction between these, start pointer option. So primary structure, when these these are a secondary structure which are a backbone for the protein structure. So when we join this, then there is a formation of a tertiary structure. So that joining between these alpha helix and beta sheet, they are called as a loop region, which are flexible one. They are not part of a backbone of a protein structure. And these loop regions are generally useful for binding of a drug molecule. See, this is the loop region. Okay, so there may be a chances of a flexibility. So then after that, the 3D tertiary structure, it is a full three-dimensional structure that is folding, where the folding of a single polypeptide chain happen, and the quaternary structure, which is the last one, as I say, which is assembly of multiple such chains into the functioning complex. So that is called as a protein structure. So if you, this is a basic things related to actually the computer-aided drug design. Then after that, in computer-aided drug design, again the thing is that which are the different types of a protein available in the research. Means if you are going for any type of a molecular docking studies or any type of a computer-aided drug design process, so there may be a chances of a different types of a proteins are there, because of their function. There is a chance of interaction happening between the drug and protein molecule. So if you are aware about this type of proteins, then proteins are nothing but a targets. Okay, so if you are aware about your target, then you can definitely get an idea about how to design your molecule. So there are 7 different types of protein structures are available, which contains hormones, immune system, then membrane proteins, storage protein, enzymes, then structural protein, plasma. So these are different types of proteins which we can target as a drug target. Then after that, the computer-aided drug design approach, where we can apply these things. So if you have a ligand structure, sorry, if you are unknown about your ligand as well as a protein structure, target means nothing but a receptor, okay, and ligand means nothing but it is a drug molecule. So every type of a ligand not may be a drug, but every drug molecule can be a ligand. Ligand is nothing but a chemical entity. Either it should have the drug like properties or not. Okay, sometimes it may get fail. So if the ligand and protein both are unknown, then you can go with the combinatorial library as well as a high throughput screening. Then after that, if the ligand is known, you have the ligand molecule, but your protein structure is unknown, then you go for virtual screening of all the targets which are available in the human genome, and then you can decide on which particular target your drug is going to bind. Then after that, QSAR is another approach which is a emerging one, and there are many types of things are coming under the banner of a QSAR. So which is a actually it is a it is very important area in the computer-aided drug design right away. Then pharmacophore modeling, another approach. If you have a ligand molecule, then pharmacophore you can generate. So that pharmacophore is nothing but that is a chemical, that particular chemical having different functional regions, means hydrophobic region, hydrophilic region. So on the basis of that, you can design your molecule. After that, if the ligand is known and target is known, then you can go with the structure-based drug design, ligand contacts, then uh, uh, ligand bound morphing. So these are the some different techniques that you can apply. And if your ligand is an unknown and target is known, then you go with the de novo drug design, virtual screening based on target as well as a ligand. So these are the some techniques that you can apply in the computer-aided drug design. So why I am going to tell you about the computer-aided drug design, you will get to know in this image. So if you see that, see here, the diagram that predicts the overall process of a computer-aided drug design. See here, this is the protein structure which is isolated, which is generated on the basis of a homology modeling. So what is a homology modeling? It is an artificial method for designing, for generating the protein structure. But in actual, what generally the scientists do for the protein structure prediction, so they are giving the drug to the individual, and then after that, after ingestion of a drug molecule, that particular target can be removed, generally removed from the individual, and that kept into the path of this X-ray crystallography. So after that, what the X-ray do? X-ray identify each and XYZ coordinate of each and every element present in the amino acid which belongs to that residue which is present in the protein structure. So that each and that whole information is coded into the text file that having the extension as a PDB file. So whatever the PDB we are getting from RCSB, that is based upon the PDB file that is getting from actual experimentation. So RCSB having PDB file which is getting from actual experimentation, and the homology model means what? The model, the protein structure which is generated due to by artificially it is generated, means if you have a sequence of a amino acid, so on the basis of that you can generate the protein structure by taking the different templates for the particular amino acid sequences present in the protein structure. So on the basis of that we are getting the protein structure, we are joining them, and then we are getting the new PDB file which is get minimized and used for the further purposes. So this is the protein structure which we are getting, and after that, that protein structure is used for structure-based as well as a ligand-based drug design processes. So structure-based drug design and ligand-based drug design, these are the two methods that we are using for the drug development process. Okay, so in the drug development, structure-based means what? It is a protein-based, it is a receptor-based drug design. Ligand-based means what? It is a drug-based drug design approach. So here, in the structure-based and drug design approach, ligand-based drug design approach, the last step is nothing but molecular docking. So for every type of a computer-aided drug design process, there is a start as well as an end can be done by the molecular docking. So that's why if you are aware about the molecular docking, then there are near about 50% chances that you will get a novel compound, and you be the part of every type of a computer-aided drug design if you are aware about the molecular docking. So just start with the molecular docking, and then you can ruin into the computer-aided drug design processes based upon different things. Then after that, the definition of a molecular docking. So molecular docking is nothing but it is a complex between macromolecule and micromolecule. So micromolecule is nothing but a ligand, and receptor is a micromolecule is a ligand, and macromolecule is a receptor. So due to the molecular docking, we are getting the different poses between the protein and ligand, and that is nothing but a protein ligand complex, and this complex gives us the scoring. So every type of a molecular docking software, they having a different types of a scoring functions. So on the basis of that, we are getting to know that which protein ligand complex has a minimum binding energy. So this type of things are generally happening into the molecular docking studies. Then after that, what the molecular docking is doing in a molecular docking, we attempt to find the more probable binding conformation of two molecules which minimize the energy through the maximize the interaction. The extension of a molecular modeling approaches goal is to find stable complex that I already discussed. So why to do the docking? So to generally the lead compound that should show the biological activity, which should be novel one and has a potential to being structurally modified for the improved biological activity, selectivity, and drugability. Obviously, molecular docking does not give us idea about the biological activity of a molecule, but we can identify the affinity of a drug molecule with the protein structure, so that on the basis of that we can find out either there is a possible interaction happening in between protein and ligand or not, that we get. Generally, even we can't predict that either the drug is agonist or antagonist, but if you are aware about the affinity of a drug molecule, then definitely you will get to know that that particular compound has a affinity with the receptor at the particular site, and on the basis of that we can determine its affinity. We can get idea about either it is a druggable or not. So after that, so this is a typical docking workflow. First, there is a target selection and ligand selection. Both these processes are simultaneously should be run, so that to minimize the time required for the molecular docking. So target selection, after that target preparation, and then ligand selection and ligand preparation. So target selection and ligand selection, these two methods are generally based upon different approaches, based upon your approach, your scientific approach. If you have a particular approach, means for example, if you have a particular phytoconstituent, so you have to know that your compound is a if you have that particular phytoconstituent, and if you want to find out that the activity of a particular ligand onto the target molecule, then you have to see, for example, it is a anti-TB agent, it is there. So you have to find out the different anti-TB targets simultaneously, and then you can dock this ligand onto different targets which are available in the path of a bacteria, that Mycobacterium tuberculosis bacteria. So whatever the target available in their body, if the drug is showing proper affinity with that particular receptor, then definitely your drug is a effective as an anti-TB agent. Okay, so like that you have to find the target, and you can, if how to select the ligand and everything I have discussed in the next slide. Then target preparation and ligand preparation, how to do that, I already done some detailed things related to this. Then after that the docking, and after that analyzing the results which we are getting after the docking process. After that, the target selection. So how to select the target? So these are the 6 different things which are related to the target selection. First is these are very important things that you have to keep in the mind during the target selection. So how to select the target? We are actually doing that practically. We are going for that. But before going to that, I want to mention some things that are generally skipped for the molecular docking. Actually, molecular docking is not an operator job. You have to think in a multiple ways so that you will get a proper target for the desired study. So first is the drugability, first is the biological relevance. Generally, it is the most important factor to consider in the biological relevance of a target. The target should be associated with the disease or condition you are trying to treat. The link between this target and disease should be well established in the scientific literature. For example, if you have a working on a cancer, you might select a protein that is overexpressed or mutated in the cancerous cell. So generally, anti-cancer agent, anti-cancer protein structure, there may be a that should be mutated one, because in cancer mutation happen, and that's why the overexpression happens. So that's why it should be mutated. Then after that, the drugability. So the target should be druggable, meaning it means that it should have a regions where drug could feasibly bind and exert an effect. Then proteins with a deep and well-defined pockets are generally often considered good target for the selection. Generally, surface proteins are surface targets are not generally used for the binding studies. Okay, if your target is a deep into the protein structure, then it is generally preferred. Okay, the drugability of a target can often be predicted using the computational tools. There are many ways that we can use this, by which we can use this, find this drugability. Then the structural information. So for docking studies, it is crucial to have a detailed 3D structure information about the target. So this information is usually obtained from the technique like X-ray, NMR. Nowadays, there is a electron microscopy is also coming, so which can resolve your molecule very deeply. So and that can be found in a database like Protein Data Bank. Okay, the more information you have about the target structure, especially the binding site, and the more likely you if you are aware about it more prominently, then definitely your docking study will be successful. Then the selectivity. The target should be selective to avoid the off-target effect that could lead to the side effects. It is essential to consider the similarity of a target to other protein in the human body. So then after that, the existing ligands. So it is generally helpful to know if there are already known ligands that bind to the target. So this can be served as a starting point for the design of a new drug molecule, and that can provide a valuable information about the type of chemical entities that target is likely to bind. Then after that, the last one is assay availability. So you will need to verify the result of your docking studies with the biological assays. So it's helpful if there are already established assay for your target or not. So these things you have to keep in mind. After that, the selection of a ligand. The selection of a ligand for molecular docking is an important step in the computer-aided drug design process. So some consideration like the first one is a chemical diversity. So having a diverse set of a ligand can increase the chances of finding a hit, and high throughput screening methods oftenly use for the large libraries of a diverse compounds. Then after that, the similarity to known binders. If there are already known active compounds against your target, you can select the ligands that are structurally similar to these compounds. So this can be done using various chem informatics tools. Okay, so like uh, similarity, known binder, co-crystal ligand in chem informatics tool. Uh, there are many databases are available on the basis. Then SciFinder it is there. Uh, then React it is there. So we can find the similarity between the known binder, so that we can design the new molecule which having the similar contact, similar structural parameters that are matching with the already bound compound. Then drug likeness. So consider the selecting ligand that possesses the drug-like properties often defined by the rule of Lipinski, rule of 5. So these rules suggest that the good drug should not be too large, less than 500. It should be less than 500 Dalton. Should not be too lipophilic. It should have a log P less than 5. Should have less than 5 hydrogen bond donors. Then less than 10 hydrogen bond acceptor. So all these parameters are generally available in the databases right away. Before that, we have to apply these filters, and then we have to screen the we have to go for virtual screening. But right now, all the databases having all these provisions. Then after that, the ADMET toxicity prediction. So you may want to consider absorption, distribution, metabolism, excretion, toxicity properties of a potential drug before going to molecular docking. So some computational tool that can predict these properties and that can help you to select the ligand that are more likely to have a good pharmacokinetic parameters and few side effects. Then after that, the availability. If you have to plan the experimentally test compounds identified through the docking, consider the availability and cost of potential of that ligands. It's often practical to select these compounds that are readily available from the chemical vendors, as well as sometimes we can apply the this synthetic accessibility score, which is already available with the many tools like that of simulation plus ADMET predictor. It contains this. Then again Swiss ADME, it has this score. So if your score is below 6, then your compound is a synthesizable one. So this type of things you have to apply before going to the virtual screening as well as the molecular docking purposes. Then after that, the chemical space. So depending on the target, you may want to explore the different areas of a chemical space. For example, if the previous attempt to target protein has failed, and you may want to select the ligand that are structurally different from those previously attempted. So remember that the ultimate goal of selecting ligand for the docking is to identify the compound that will bind to the target protein and modify its activity in a way that provide a therapeutic benefit. So therefore, while these all considering all these factors that can guide you, the ligand selection and the process is often interactive and require refinement based on the result of a docking studies and subsequent experimental validation also. Then protein preparation. So these are 7 parameters that you have to consider during protein preparation. So first is a structural source. So retrieve the 3D structure of your protein of interest, which is actually obtained from the experiment. So this data is often obtained from the protein data bank, from the RCSB, and the database that collect the 3D structure of a large molecule such as a protein, nucleic acid, and enzymes, etc. So the selection should be based on the resolution. There are many parameters, but simultaneously I will tell you the resolution is the first parameter that can be applicable. the resolution should be lower, and then after that, validation parameters. There are many validation parameters. Whenever we are going for actual experiment, actual operation, I will narrate you the different things that you have to consider for the protein preparation. Then after that, the cleaning of a protein. So you have to clean up the downloaded protein by removing the water molecule. Whatever the non-standard residues are present in your protein, that you have to remove. Then the ligands which are not generally involved in the binding pocket, and after that, any other non-standard protein molecule like glycol, then ADP, AMP, ATP. So any type of a non-standard residue, if it is present, hemoglobin. So if they are not present in a binding pocket, you have to remove that. Water molecule, that you have to remove. However, if any ions or small molecule are crucial for the protein structure or function, they should be kept. Means if the binding pocket contains a means your drug with that of a, for example, sometimes there is a hemoglobin is present in the binding pocket with that of a ligand molecule. So actually the role of hemoglobin is as a carrier for that drug molecule. So you have to keep that hemoglobin as it is during docking studies, and you have to go for simultaneous docking. So you get to know that which type of a docking you have to apply for this process. So cleaning is a crucial step. So if binding pocket contains multiple ligand, then you have to keep them except water molecule. Water molecule you have to remove. Again, there is a hydrated docking is one of the part in which you can keep the good water molecules which are available in the binding pocket. Okay, so there are many tools that can perform this automatic cleaning, like that of a Schrodinger's glide. So that can identify all these non-standard residues, but still the human sense is mattering a lot. So if you have a good sense of humor for the molecular docking, then definitely you can get a proper, you will get to know that how to apply your knowledge into the molecular docking. So adding hydrogen, so X-ray crystallography doesn't identify the hydrogen molecule. So whatever the hydrogen molecule present, hydrogen has a molecular weight only one. So that's why X-ray crystallography doesn't identify the atoms which having a one atomic weight, and that is the only one, that is a hydrogen. So if if hydrogens are not identified the X-ray crystallographic structure, then if your structure is a X-ray result, then you have to add it, and you have to see if you add hydrogen, then definitely they can add in the any direction. So that's why you have to, after addition of these hydrogens, you have to minimize them. Okay, so for NMR is identifying the hydrogens only. So that's why if the NMR structure it is there, there is no need to add hydrogen. So depending upon the resolution method, you have to go with the addition of hydrogens. So hydrogen atom needed to be added in silico, considering while protonation state of a protein is also mattering a lot. If during dealing with this addition of a hydrogen, for example, anti-cancer drug therapy, it is there. So in that case, your protein is acidic in the nature, because the environment is acidic. Even in anti-inflammatory case, design of an anti-inflammatory agent. So your cyclooxygenase has an acidic nature. So that's why you have to go with the literature, and you have to find out what is the pH of that particular environment for that protein, and then according to that you have to minimize, you have to apply the pH parameter, and then you can go with the addition of a hydrogen. So this is a proper docking. Okay, so whenever you are going to write a good paper, then the reviewer is going with all these things, and if you are writing like a way, definitely he get to know that you are a good researcher. And if you are dealing with all these things during writing the paper, see, docking, doing docking is not a big task. Right away many people are doing, but if you are the major thing is that how to write the literature, how to write a research paper. So if during dealing with this research study, If you are considering all these things in the mind, then definitely you will get a good publication. So dealing with the missing residue. So if there are any missing residue in the protein structure, you may need to know, then to add them using the homology. See, addition of residue, it is done by only homology modeling. See, for example, why there is a missing residue? So many organisms, depending upon their environment, so there are any protein molecule can be generated by the two types of residues. One is a essential and other is a non-essential residues. So non-essential residues can be generated by the body, and essential can be taken up by the diet. So if their diet doesn't contain that particular amino acid, then that our body, our RNA doesn't get stopped at that step. RNA skip that amino acid and then proceed for the further amino acid and complete that protein and get ruled that process. So work can't get stopped. So that's why such, or sometimes what happen during isolation, there may be a chances of a miss or resolution process. There may be a chances of a unidentified some residues. So that can be added into the protein structure. So in that case, if any residue which is present in the binding site, then that you have to add by using homology modeling. There is only tool, that is the only tool which we can add the missing residues. Missing atoms we can add by any tool which are free, but missing residues can be added by only two tools. One is a modeler, and means that is a homology modeling approaches by using a modeler, or second tool is that Schrodinger's glide. But in that case, your sequence should be present in that protein. If you are cleaning before the protein preparation, then and there are if the genome sequence is cleared before the protein preparation, then again that protein can't get identified which particular amino acid is missing. If the sequence is present in that file, then definitely it can carry from somewhere and it will get added into the protein structure and complete the missing residues. So these are the things you have to keep in mind while dealing with the protein preparation. Then after that, the charges and energy minimization. So this is very important thing for the protein. See, many people are saying that our docking study is a different and our result, actual biological activity results are the different. So this can't be happening. And what to do for this should not be happen? So for that, you have to minimize your protein properly so that you will get your results should be matched with the actual biological activity. So whatever the protein we are isolating from the body, it will get into the strain state. So that's why while resolving into the X-ray crystallography, our protein is at the strain state. So in that state, we have to resolving our protein structure, and that is coded into the text file. So during protein preparation, we have to minimize them so that we get a proper protein structure which is ideal, like that of a resting stage which is present in our body. Okay, so to assign the correct charges to the protein residue, considering their ionization state at a physiological pH, so afterward an energy minimization step is usually preferred to relieve any steric clashes or steric strain in the protein structure. So this process helps to achieve the low energy stable conformation of a protein structure. So there are many force fields that are used for the energy minimization, which right away there is a best force field available for the protein structure, that is opals, which is developed by the Schrodinger. So I am forcefully telling you about the Schrodinger because right away they have developed very good things, but it is paid. But rather than that, there are many other things, many other processes, many other force fields are available which are free which are just similar to that. That is nothing but a molecular mechanics force field, which are also freely available, and we can apply them into the energy minimization of a protein by using the autodock. Okay, that is also a better tool which is used for the minimization of a protein. See here again, I want to mention that. So minimization of a protein and minimization of a ligand, these two things are generally based upon their structural characteristics. See, for example, protein structure is a bigger one. So that's why for the minimization of this protein structure, we are using a molecular mechanics force field which contains more approximation. See, minimization is nothing but a calculation. It is a statistics. So if during minimization, there are many calculations are happening in the background. So that background processes, background calculation, if you want to minimize, then you have to go for more approximation. And on the basis of that, we will get to know that our protein, how to minimize the protein and how to minimize the ligand. So there are two types of a force field. That is, first is a molecular mechanics force field, and second is a quantum mechanics force field. So for protein structure, you have to use a molecular mechanics force field to minimize, and because it contains more approximation, but the protein structure is a more bigger one. It has a molecular weight near about 2 lakh to 4 lakh, and that's why it may teases to means there is a need to short period to minimize that protein molecule. Okay, and if we apply the quantum mechanics, then for molecule having molecular weight 200 to 300, it takes near about 2 days to minimize in a normal laptop. So that's why, see, its molecular weight is below 300, still it is taking this much of a time. So how much it will take for protein structure? So that's why we are using a quantum mechanics force field to minimize the ligand molecule, not for the protein molecule. But in quantum mechanics, we get to know that actual minimization, and there are actual calculations are doing, and there is a less approximations are happening. So that's why quantum mechanics minimized structure gives you proper result which are matching with the biological activity. So during docking, if you go with the quantum minimization of a ligand with the quantum mechanics and molecular mechanics force field for the minimization of a protein structure, definitely your result will matches with the biological activity. Then the sixth step is defining the active site. So there are many tools. I will narrate you how to find the active site. So if your active site of a binding pocket is known, then it should be defined with the software, and there are many algorithms to predict the binding site. So there are two types of proteins can be chosen. First is the protein which, see, if you have a available protein with the drug molecule or co-crystal ligand, then you have to choose that one. Otherwise, if your protein is a crude one, virgin one, then that protein the grid pocket for that protein can be identified by using a CASP and different meta pocket, and different types of tools are there. So we are going to identify their binding pocket. So binding pocket is nothing but a bigger white space in the protein. Okay, and the next step is preparing protein grid. So grid pocket. So before docking, the grid is prepared around the active site. The grid dimension should be large enough to encompass to enter active site. The grid is what the software uses to map the potential interaction sites with the ligand. So each docking software may have slightly different preparation protocols, and users should refer to specific software manual or tutorials to exact step. So for example, popular molecular docking software and grid finding pocket, Autodock, Dock, then Gold, MOI. So these are the different types of tools that are used for this protein preparation. The next is the ligand preparation. So just like that of a protein preparation, ligand preparation is also a vital step in the molecular docking process. So the first step is a ligand source. So if you are collecting your molecule in 2D or 3D structure, then if they are downloaded from the different databases. Generally, if you are, then in that case, if the database is a bigger one, for example, it is of a high GB, then in that case you can download it into the 1D format, and 1D file format is nothing but a smiles file format. So which are the different file formats are there, I will tell you in this workshop also. So so if 2D 3D are there, then most preferentially you can go with the 3D structure. If 3D structures are present, go with that. If 1D and 2D present, then go with the 2D. Okay, so likewise you have to go based upon size also. If the size is bigger one, yeah, and many times it is happening that the database can't be downloaded. So in that case, you can download the text file format or smiles file format. So it can, and then you go for 2D and 3D conversion of these molecules. Then format conversion. So convert downloaded ligand molecule into the file format that run by the particular docking software. Here in this workshop, we are dealing with the Autodoc Vina. So that's why we are dealing with the MOL2, PDB, SDF. These are the file formats, and the running file format for this Autodoc Vina, that is a PDBQT, which is a charged PDB file. So that's why we are dealing with this file format conversion by using Open Babel. Then after that, the adding hydrogen. Again, the source of a ligand matters a lot. If the ligand structure doesn't contain hydrogen, you have to add them and then minimize your compound. Then protonation state. So depending upon the availability of a ligand or its environment, you have to minimize them, you have to protonate them accordingly. See, for example, basic compound, it is there and it has a ionization state into the stomach. Then in that case, you have to first of all convert it into the acidic state, and then you have to dock that protein. Because if your drug is a basic in the nature, then definitely it will get to neutralize due to the stomach acid. And that's why you have to consider all these things, all the environment which is available in our body. Then after that, the conformer generation, tautomer generation. So most of the time, the metabolism is most prominently convert your molecule into the tautomeric form. Means, for example, keto to enol, enol to keto. So these are generally happening. So you have to consider the tautomeric form of a drug also. So after 2004, the WHO is not going to deal with the only one conformer of a compound. It is dealing with both the conformer. So if your compound, any particular compound having R conformer is active and S conformer is inactive, then such type of a drug can't be approved by the WHO. So that's why we have to deal with the tautomers also, and what is the effect of these tautomers onto the particular receptor, that also can be written into the manuscript whenever you are going for the particular research study, docking study. Then generating the 3D conformation. So if you have started with the 2D structure, you will need to generate their 3D conformation. So there are various tools which are used for this purpose, that is a Corina, Open Babel. So there are different types of tools that we can use for conformer generation. R conformation, it is there, then you have to convert them into the AS. So these are generally considered. Then after that, generating the multiple conformation. Then energy minimization. So energy minimization you have to done by using the quantum mechanics, or there is another method for minimization. So that is nothing but what you can say semi-empirical force field. Okay, so semi-empirical force field is a blend of this molecular mechanics and quantum mechanics, and both we are using for minimization of a ligand. So that is a better one, and it is also less time consuming rather than quantum mechanics and greater than that of a molecular mechanics. Then dissolution. So if the ligands are dissolved, solvated, or surrounded by the water molecule, so these will typically be removed prior to the docking. So exact procedure can vary depending upon the software. Such if you are using, so if you are using that, so it is important to refer to specific tutorial means manual for before using any particular molecular docking tools or softwares. So so all these things we can add manually for the Autodoc Vina. The next is what is docking? In the docking, there are, see, after ligand preparation and protein preparation, actual docking process it is there. So I'm not going to in depth about the docking procedure. So there are background calculations are there on the basis of a different docking algorithm. There are different types of tools are there. So there are two types of algorithm. One is a deterministic and second is a stochastic. So on the basis of that, there are different algorithms are used by the different tools, and based on that, the tools are determining the poses of a molecule in the binding pocket. So and then we got to know that which score. So you can't be predict, we can't be compare the scores of two protein ligand complexes by using two different tools. So you have to use only one tool to compare the scores of binding energy scores of docking studies. So these are the sampling algorithms. So different types of tools are using these algorithms. Say for example, if you, this is an older one. Actually, this article is published in 2017, and during that, if you see the Vina is using genetic algorithm. See here, Autodoc. Autodoc is using a genetic algorithm, which is a stochastic approach for the scoring function. Okay. Then different these are the popular docking algorithms, so which are free in Autodoc, Autodoc Vina, Doc, then Argus Lab, Swiss Doc. These are the free tools which are used for the docking purposes. Then commercial are ICM Pro, ICM, then Glide, Gold, Flex, Moligro. Then protein docking for that plus, these are free. Plus Pro, Hex, Haddock, ZDoc, all these are the free. Generally, if most of the peoples are from pharma background, so that's why we have to go with the protein ligand interaction rather than protein interaction. So if there may be having antigen antibody interaction, then you can go with the this, and after that you actually this is a very simple docking. Okay, then which are the different types of docking? So I am explaining this and then directly jump to the actual experimentation. So the types of docking. So this is a rigid docking, then flexible docking, protein docking, protein ligand docking, local docking, global docking, bound and unbound docking, then covalent docking, heterotic docking, metalloprotein docking, inverse docking, virtual screening by docking. So what are these? So first is a rigid docking. Rigid docking is nothing but where we are considering the protein as a rigid and ligand as a flexible. Then flexible docking, the both protein and ligand both should be considered as a flexible, and that is called as a inducer fit docking. Okay, but it is not a free, it requires there is only one tool that can be done, that can be used to do by all these dockings by the Autodoc Vina. Okay, then protein docking that can be done, this is one of the type. Then in that antigen antibody, then protein antibody, protein and DNA docking. So such type of things are generally doing under the banner of this protein docking. Then local docking and global docking. So local docking means what? We are going to find the particular active site and then we are going for um, uh, mm, docking at the particular area. And global docking means what? It is a blind docking. Whole protein we are considering, and we are going to dock our ligand onto that protein molecule. Then after that, bound and unbound docking. Covalent docking is another type. Then hydrated docking is another type. Hydrated docking means what? It contains um, actually we are finding the good water molecule which are, see, in unbound state the protein void space is filled with the water molecule. So when our ligand molecule come in contact with the protein structure, then the drug has a capacity to repel that water molecule present in the void spaces in the protein. So after that, again, there are some spaces where the complementary binding sites are not present. So that's why the water can make these contacts between the protein and ligand. So that protein, that water molecule is called as a good water, and that can be considered during docking study. So that type of a docking is called as a hydrated docking. Then after that, metalloprotein docking. So if the metal is present in the binding site, then that type of a docking is called as a metalloprotein docking. Then inverse docking. So inverse docking means what? You have only one compound, and you have to dock that with the multiple protein at a time. So that is called as a inverse docking. Then virtual screening. So in that, many molecules at a time we are doing docking. Then inducer feed docking, I already told. Metal ion docking. So again, coordinate docking. So here you can see that there is a metal ion is present in the binding site, and that you have to consider for the interaction for the protein ligand interaction, protein ligand docking. So if you see, there is a one example I am telling you that right away, right now there are many tools are used. Many work is doing on COVID-19, and the COVID most populated popular target is protease. So if we see the actual structure of this proteases, then it contains a zinc in the binding site. So if that zinc is present in binding site, then we have to consider that during docking study. So near about 90 to 99% people are removing that zinc molecule and doing docking. So that type of things should not be done during docking studies. This you have to consider, and you have to go with that. That is called as a metal ion docking. And after that, these are the various tools which are used for the docking studies. So this is an older image in which the AutoDock is generally used by many people, as its algorithms are very good, and second most is a gold that time. Then after that, glide is one. So here it is not given, but right away glide is used by the 50% peoples. Then after that, near about 25% peoples are using AutoDock, and then the remaining are in the 25%. Most of the peoples are using this glide, Schrodinger's glide, and AutoDock Vena for the docking purpose. So what are the important factors for considering the docking software? First is the sensitivity on the transferability of a parameter, including the starting information, then its adaptability to additional scoring function, pre and post docking processes and filters, then ability for interactively refining the docking procedures, protocols based on a new result, then the design components and result validation studies, then speed, then its customer support and cost, then code availability. So most of the docking procedures are generally doing on on the basis of a script. So that scripts also should be available then there should be upgrading facility should be available. So all these parameters we have to consider while purchasing the docking software. Actually, for academics, many of these tools are generally free. So that's why there is no need to buy any tool for the docking process if you are a master in all these things. If you are aware about all the things, then there is no need. You can till this time I have not bought any type of a tool for the docking process, but I have published near about 20 research paper in a good impact factor journal on the basis of Autodoc Vina. Then after that, the challenges for the docking approach. First is the binding affinity is only one of many attribute of a drug. Binding affinity, this should be one attribute. Then protein structure of a most druggable target undetermined. Then these are the challenges. Okay. Then identification of a binding site by the docking software. This is again a bigger approach, because many times what happen actually, which is a particular binding site, if it is not, if there is a no any co-stress ligand is present, so that can't be identify the docking tools. Then after that, the dependencies on the protein and ligand structure, their source, then flexibility, starting conformation, structure diversity, protein state, everything. Then similar ligands may unexpectedly bind to quite different modes. That may happen, and favor larger and more complicated molecules can't be synthesized. So this type of challenges are happening after the end of a molecular docking. So we have to consider all these things. These are the some example for the molecular docking process. So examples are many: antiviral agent, then anti-cancer agents. And at the end of this introductory part, I want to tell you that artificial intelligence. So artificial intelligence, most probably we can apply in the different processes to modify the process, which is means whatever the parameters we have studied. Means, for example, protein preparation, ligand preparation, post prediction, scoring function. So there in scoring function we can apply this artificial intelligence like machine learning technologies, so that we will get to know that the advanced version or advanced score on the basis of a previous scores. So these are the things we can apply to get a better docking process. Now we are actually moving to the docking. Okay, so these are the steps in the docking way, which I already told: protein preparation, ligand preparation, then active site prediction, then grid preparation, and performing docking using Autodoc Vina, and analysis of the result. So here Autodoc tools are generally, see, I'm using Autodoc Vina. So for that, I'm going by this route only. Okay, so this is for Autodoc. This is the previous slide that I use for the Autodoc tools, which is a graphical user interface that is run by the run on Windows. Okay, so first of all, what are the protein you are getting and ligand you are selecting, then that you have to convert it into the PDBQT file format, and then finding the grid molecule, and then directly go for the docking. So this is a process for Autodoc Win. Now, which are the different file formats which are generally we can use for this workshop? I will tell you. See, so this is a open Babel graphical user interface, which is wait. Okay. My screen is visible to all of you?